Hello, everyone, and welcome to this Integrated DNA Technologies webinar, Intro to rhAmpSeq. My name is Ginny, and I will be serving as moderator for today's presentation. Today's presentation will be given by Simon Dunbar, APAC NGS Field Applications Manager. Simon is a member of the IDT NGS Field Applications Group, providing support to researchers using IDT's range of NGS solutions in the field. Simon completed his bachelor's degree in biomedical sciences with honors at University of Ulster. Following that, with postgraduate studies at Queen's University Belfast, where he earned a master's degree in molecular biology. Prior to working at IDT, Simon investigated the therapeutic potential of siRNA for treatment of conditions caused by dominant negative mutations. He has been with IDT for 5 years. Simon's presentation should last around 30 minutes, and following the presentation, he will answer as many questions as possible from attendees. The question and answer session will be conducted by myself. As attendees, you have been muted, but we encourage you to ask questions or make comments at any time during or after the presentation by entering your question in the Q&A box. Also, please note that you can expand the presentation slide window for easier viewing. In case you need to leave early or want to revisit this webinar, we are recording the presentation and we'll make the link to the recording available on our website a few days after the presentation. We will also post the recorded presentation on our YouTube and Vimeo channels. You will receive links to these in a follow-up email. So now, let me hand this over to Simon for his presentation.
Thank you, Ginny. G'day everyone, thanks for dialing in to today's presentation in rhAmpSeq targeted amplicon sequencing. During today's presentation, we will cover targeted sequencing at IDT, the underlying chemistry of rhAMP technology. Then we'll look in detail at the rhAmpSeq system, cover some protocol options, design, key benefits, and finally we'll finish off with some example data and applications. This should only last around 20 to 25 minutes. This is a quick overview of IDT's involvement in next generation sequencing and targeted sequencing. So if we follow the top line here, the top blue line, you can see that we have a general use library prep kit known as our Lotus and XGen adapters. So this can be used for whole genome sequencing, or you can continue to follow this blue line and proceed to hybrid capture using IDT's range of XGen enrichment products. Following the bottom blue line, you can see we end up at rhAmpSeq amplicon sequencing system, and this will be the focus of today's presentation. Before I dive into further detail into rhAmpSeq and rhAMPCR, I want to spend a moment looking at the differences between hybrid capture and amplicon sequencing, and also the pros and cons of each technology. On the left-hand side of the screen, you can see an image of someone fishing, and that's a good analogy for hybrid capture. Essentially, we are pulling out our region of interest from a sample using a biotinylated DNA oligo, which is complementary to the target and often referred to as a bait. So we use the bait to fish out our favorite gene, as indicated in this little image here. So moving the advantages of running capture is that it's a more accurate representation of your original DNA sample. It's better for larger target spaces. Because we perform adapter ligation during library preparation, we can build in error correction through the addition of UMIs. And generally, we get a higher degree of uniformity with hybrid capture. Some of the disadvantages of hybrid capture in that the protocol is pretty long, usually a full, IDT is one of the shorter protocols, lasts around 9 hours. There are variable results for smaller panels under 1,000 probes. And if you're only running one capture per sample, then it can be more expensive. On the right-hand side, we can see a very simplistic overview of amplicon sequencing. And again, here we're targeting specific regions of interest, but instead of using bait oligos and pull down, we use locus-specific primer pairs to enrich for our regions of interest. So there are some advantages to using amplicon sequencing. The workflow is very easy. It's just 2 PCRs, so there are fewer steps. There's no need for DNA shearing. It's better suited to smaller target panels. Again, because it is just 2 PCRs, it is very easy to adapt to automation. And again, because we're using a pair of primers to enrich for a particular target, it generally has a higher on-target rate. Some of the disadvantages are that ultra-low allele frequency and detection is not quite proven. We can't incorporate UMIs in amplicon sequencing just yet. Specifically to rhAmpSeq, it's for profiling indels and SNPs, so we can't support a gene tiling coverage right now. And also there are a limited number of primers that you can have in a single reaction. Right now we're topping out at 5,000 amplicons, so 10,000 primers. So what is the rhAmpSeq system? So it's a novel, innovative suite of reagents for targeted amplicon sequencing on Illumina NGS platform. And the technology is based off IDT's rhAmpPCR chemistry. And the benefits of rhAmpPCR are that it allows to perform high levels of multiplexing. It's a simplified workflow with only 2 PCR steps, and this enables accurate targeted sequencing results with high on-target rate and uniformity. So what is rhAmpPCR? If you've known IDT for a while, you've probably come across rhAmpPCR, formerly known as rhPCR. And this is a technology that was developed at IDT, which is more specific than standard PCR. This is achieved by exploiting the unique properties of the enzyme RNase H2. This enzyme recognizes DNA-RNA heteroduplexes and cleaves at that site. So we've exploited this property to develop primers, which are blocked and have a single RNA base near the 3' end of each primer. So we can see here on this schematic exactly how that property is translated into a more specific PCR. At the top of the image here, you can see rhAmpPCR primers, both forward and reverse, and they have an RNA base followed by a blocking group. Under normal PCR conditions, these primers are blocked and not extendable, so this results in no PCR product. It's only when we add RNase H2 that the DNA-RNA hybrid is recognized and cleavage occurs, and this removes the 3' blocker, exposing a free hydroxyl group, which can then be extended by Taq polymerase, and this generates our PCR product. So RNase H2 is sensitive to mismatches around the RNA base, so unless this primer is perfectly bound to its target, we don't see any cleavage. So again, why rhAmpPCR? And as I've just mentioned, RNase H2 is sensitive to mismatches around the cleavage site. So this enables higher specificity, gives us a lower potential for primer dimers. That allows us to perform a higher level of multiplexing, have modular panel designs, and flexible amplicon sizes. This is a snapshot of this in action. Here we can see 2 gel images. The left gel image is of a 96 amplicon panel. So the same panel has been run here with standard PCR primers and also rhAmpPCR primers. Looking at the standard PCR primers, here we can see that they're generating large amounts of primer dimers and very little of our desired product. However, the small panel, which is the same panel, just run as rhAmpPCR primers, shows almost no primer dimers. The gel image on the right-hand side shows 2 larger panels run just as rhAmpPCR panels, and these are both close to 1,000 targets. It's a similar story here. We can see that there are very little primer dimers, and we're generating most of our desired product. So this chemistry enables very high levels of multiplexing, and this reduces the complexity of our amplicon sequencing system. It means we have fewer steps in our protocol. If you're interested in learning more about rhAmpPCR, we go into greater detail in this 2011 paper from IDT's research group, and this describes the chemistry underlying rhAmpPCR, and this is available on our website. You can download it for free. So as I mentioned, we've combined rhAmpPCR and amplicon sequencing to create rhAmpSeq, a targeted amplicon sequencing technology. By using rhAmpPCR chemistry for amplicon sequencing, we can overcome the challenges associated with extremely high levels of multiplexing. On the right-hand side of this slide, you'll see how we combine rhAmpPCR to create rhAmpSeq. And if any of you are familiar with amplicon sequencing technology, you will notice this is very similar to others in that it's just 2 rounds of PCR to generate Illumina-ready libraries. PCR1, known here as targeted rhAmpPCR, this is where we use our target locus-specific rhAmpPCR primers. So when the locus-specific rhAmpPCR primers find their target, we get cleavage of the blocking group, which allows generation of our rhAmpPCR product. The rhAmpPCR product is used as the input for PCR2, and this is an indexing PCR. So during this PCR step, sample indexes and flow cell attachment arms are added to our rhAmpPCR product, and the result is a pool of sequence-ready amplicons. All of this is possible with only 3 components, which are listed here on the left-hand side. A rhAmpSeq panel, so these would be your rhAmpPCR primers targeting your regions of interest. And right now, IDT is handling all of the design for you free of charge. A rhAmpSeq library kit, these will contain the master mixes required for PCR1 and PCR2, and also the RNase H enzyme. And finally, the last component you require is rhAmpSeq indexing primers. So we currently have 96 I5 indexes and 96 I7 indexes. This allows for 96 unique dual index combinations or over 9,000 combinatorial dual index libraries. We've actually just launched stocked indexing primers this week, and they are compatible with rhAmpSeq, and they are available to be viewed on our adapter page. There are 2 versions of our rhAmpSeq protocol, standard and high throughput. The high throughput protocol has some significant workflow advantages, so I'm going to focus on this version for the rest of the presentation. So we have our input here, and it'll be between 10 and 50 nanograms. And as described previously, we perform our rhAmpPCR. After rhAmpPCR, we perform a 1 in 20 dilution of the rhAmpPCR product. And again, this is used as the input for the indexing PCR. Once you've run your indexing PCR, you pool all of your samples for multiplexing. And with our high throughput protocol, you simply pool each sample by equal volume, perform a single bead cleanup on the pool of libraries, and quantify the entire pool. If anyone has ran another amplicon sequencing technology or familiar with amplicon sequencing, you know that this is very much a streamlined protocol. Normally, you would be required to perform a bead cleanup after PCR1 for every sample. But with our high throughput protocol, as mentioned, it's a simple dilution. After PCR2, it's normal to have to perform a second bead cleanup on each sample, then quantify each individual sample and pool by concentration. However, with rhAmpSeq, we simply pool by equal volume, perform a single bead cleanup on the pool of libraries, and we quantify the pool. So if you're processing 96 samples, this equates to around 2 to 3 hours time saving. We've gone from having to perform 192 bead cleanups, so 1 bead cleanup for each sample after PCR1 and PCR2. This is now a single bead cleanup after PCR2, so we've gone from 192 cleanups to 1 cleanup. We've also gone from quantifying every library after PCR2 to quantifying just the pool of libraries. So this translates to significant time savings, but also significant savings on ancillary reagents such as Ampere beads. And this whole protocol is about 4 to 4.5 hours from start to finish, including quantification. This is rhAmpSeq library formation in greater detail with some terminology down here. So we define an assay as a pair of forward and reverse ramp primers designed to amplify a specific region, and a panel as a collection of assays. Here you can see we have our target in the center and our locus-specific ramp primers. Here is the forward and reverse. They both contain the blocking group and RNA base. And the ramp primers also have tails which allow for binding of PCR2 primers. PCR2 is done with our indexing primers, and these will also have the P5 and P7 sequences and also the I5 and I7 sample indexes. Once you've completed PCR1 and PCR2, this is how the final library looks. So we have our insert in the center, flanked by the locus-specific sequences, primer binding site, sample indexes, and flow cell attachment arms. So if you were to run this on a tape station, you would expect to see a peak of around 250 to 300 base pairs. Our default for the amplicon is between 50 and 120 bases, although we can tune this and go larger. Right now we're going up to around 200 bases, and so if you want all of the information surrounding your SNP, we can get you that, or if you're just interested in the SNP, you can reduce that to say 50 or 75 bases and minimize your sequencing costs. So I'm going to move on from the actual protocol and the description of rhAmpSeq to spend a bit of time looking at the design process and some of example data. Over the course of having launched rhAmpSeq, we've seen many different applications, and these are some of the most common that we have created designs for. So in the ag bio space, we see a lot of people using this for genotyping by sequencing. Internally, we use it a lot to assess the total editing of guide RNAs that we're evaluating. And also it can be used in human disease research. For example, tumor hotspot panels is a common application for rhAmpSeq. So the key benefit of rhAmpSeq is that we can get a very high level of multiplexing without primer dimers. This is made possible not only by the properties of the RNase H2 enzyme, but also by our primer design process. We have an in silico pooling QC, which is a neat process to predict interactions between each of the primers in the pool. So if we have fewer primer dimers, that will give us better results and makes rhAmpSeq an easier system to use. Here we have an example of the primer design process. So if we just take target 1 here for an example, we begin by performing exhaustive primer design around each target site. We then select the best primer pair for each target, and they're fed into our pooling QC pipeline. So this pooling QC checks for cross-reactivity between all of the primers in the pool to identify any potential issues. If, for example, it flagged the primer pair from the first target, we would remove that primer pair from the pool, go back and look at the next best pair of primers at that site and screen those against the pool. And this is done across all targets until we have no predicted interactions. So probably everybody who does primer design is performing step 1 where they're picking the best pair of primers, but the secret sauce really is the pooling QC, this in silico QC, and this is really what separates us apart from our competitors, as well as the ability to incorporate RAMP PCR. We have the flexibility to expand on panels you've already created by adding existing content. So if you test a panel and discover there are additional targets that you want in there, we can design additional primer sets and run the pooling QC against the added content. So we can even plate individual assays in wells, and you can choose to receive the panel like this on the right-hand side and create sub-panels, because all of these assays are predicted to play well together. So we know that you can combine these in any combination once you receive them, as long as they pass our pooling QC. Our primer design pipeline allows the use of degenerate bases. So normally you would have to avoid designing over these variable regions, but with rhAmpSeq, we can place primers anywhere in here. And this increases our design success rate, but also prevents allele dropout. I'm going to move on now and show you some data from some panels we've tested in-house. So before I do that, I just want to highlight some of the important metrics we considered when evaluating the performance of rhAmpSeq. And this first metric, total reads per sample, that specifically relates to the high throughput protocol, because we're not required to quantify our DNA input as well as the DNA at the end. So, each individual sample at the end, but you're also not required to quantify your input. So because we don't quantify the input, there could be a scenario where one sample has more input than the other. So we still want to be able to call all of our samples genotypes. So our specification for this is that as long as each sample is within a 2-fold range of reads, we can accurately call genotypes. mapped percentage, so the percent of total reads that map to the genome. Our percent on target, so these are the percent of mapped reads that contain the target region. Coverage uniformity is a very important metric, and 0.2x of the mean coverage is a common metric that we see used and we use ourselves. This is the percent of assays that have reads greater than 20% of the mean number of reads for each assay. And at the end of all this, we just want to be able to call our genotype, so we also look at our call rate. Here we are looking at the percent mapped reads on the left-hand side for two in-house panels, a panel of 282 targets and a panel of 994 targets. And we're also testing two input amounts, so 10 nanograms and 50 nanograms. This is the either end of the spectrum for the high throughput protocol. And you can see that for both panels and both input amounts, we have close to 100% map reads for the smaller panel and above 96% for the larger panel. On the right-hand side, we have the same two panels, and here we're looking at on-target percentage. So on the y-axis, you will see the input amount, and on the x-axis, you see the on-target percentage on the y-axis and the input amount on the x-axis. And you can see that for both panels and input amounts, we have over 97% on target. I say amplicon panels because we're using two specific pairs of primers. They generally always have a very high on-target percentage. So now here we're looking at coverage uniformity of the same two panels on the left-hand side of this slide. And we can see this graph here. And we can see on the y-axis, this is the percent of assays that have reads greater than 20% of the mean number of reads for each assay. And on the x-axis is again the input amount. And for 282 plex, we have over 95% of the assays that have reads greater than 20% of the mean number of reads. And this is above 85% for the larger panel. And this 85% will be our minimum guarantee that we will give. All of IDT's rhAmpSeq panels will be custom. We only have a single stock panel, which is our sample ID. So this is a benchmark that we're willing to set ourselves for anything custom at all. And that's quite unique. I don't think any other manufacturers do that. On the right-hand side here, we have data which shows the number of reads per sample at varying input amounts. So on the y-axis is the number of reads, and on the x-axis is the input amount. And it makes sense, as you would expect, that the samples with the higher input amount have more reads going to them. Although that is the case, and we can see that the higher input amounts consume more reads, we can see that this doesn't affect our call rate. So we're still able to accurately call genotypes from 10 to 50 nanograms. So that was some in-house panels that we had run looking at the development of rhAmpSeq. So I'm going to move on and give you an example of one of the main applications we use rhAmpSeq for at IDT, and it's routinely used in our CRISPR work. So we develop CRISPR genome editing products at IDT. And I'm not going to go into too much detail explaining how CRISPR works, but essentially we use guide RNAs to program a Cas9 nuclease, and this will generate double-stranded breaks at sites which match the guide RNA sequence. So this system is not perfect. There are intended on-target double-stranded breaks, but there are also unwanted off-target double-stranded breaks. And we can use rhAmpSeq to quantify the editing profile of a particular guide RNA at the on and off target sites in a single reaction simultaneously. So before I move on and show you data of what that looks like, I should mention that rhAmpSeq is not a discovery tool. It's a biased technique. And when I say it's a biased technique, is that we must know what we're looking for when we're performing rhAmpSeq. So in order to do that, we need to use an unbiased technique to inform our panel design. And the gold standard for identification of off-target sites is a method known as GuideSeq. And GuideSeq was developed by Keith Young's lab back in 2014. And during genome editing experiments, a short double-stranded oligo tag is added to the transfection mix. And this short double-stranded oligo tag is integrated at double-stranded breaks and is used as a tag for amplification and identification of off-target sites. So this is an example of a GuideSeq readout for a guide RNA targeting the androgen receptor. And you can see here that GuideSeq has identified 44 targets that should be interrogated by rhAmpSeq. So the on-target edit, which is the top one, and it has the most number of reads going to it, which is nice to see. And also the on-target edit and 43 off-target sites that have been identified. And we normally round this off with the top 10 hits from an in silico prediction tool. And this gives us our panel of 54 assays here. So targeting the on-target and 53 other sites. So these are combined, and this 54 assays are developed as rhAmpSeq assays, and this is what we use to assess the total editing of a particular guide. So while GuideSeq is a useful technique to identify off-target sites, it's not able to accurately quantify the level of editing at each site, and a large portion of the reads are noise, so you actually only use around 20% of the reads. So we see the need for a robust method to accurately quantify the level of editing at on and off-target sites. And for that, we use rhAmpSeq. So this is an example here again, a familiar picture that I showed earlier. So we've informed our rhAmpSeq design. We have performed our CRISPR experiment. We've extracted the genomic DNA, and we're now ready to run rhAmpSeq. So we use the DNA from the Cas9 edited cells as the input into rhAmpSeq and followed the protocol as I described earlier. So CRISPR panels are typically small, usually between 50 to 100 targets. So that being said, you have the ability to multiplex many samples in a single run because they are quite small. So here are the results of our rhAmpSeq run for the particular androgen receptor that I mentioned earlier. So here on the y-axis, you can see the percentage editing. And on the x-axis, we plotted the genomic locations of each edit site. And the first bar with the star above it, that is the on-target edit. And just notice that this is not a linear scale, this is log. And we can see that that has an on-target editing efficiency of 83%. And rhAmpSeq is quantitative, and people have asked how low of frequency variant we can correctly call. It's true that people want to be sure they don't see off-target variants. So like in oncology, low-frequency variants become harder to resolve because of background noise of the assay, PCR sequencing error. Researchers have claimed that they can resolve variants down to 0.2% with amplicon sequencing technologies. And we've seen for some target sites that possible, but for others, it's not. For other sites, we see background noise as high as 7%. So we recommend setting our background noise level at 1%, and we're currently looking at ways to reduce this. So, I mean, it may be possible that there are events happening down here and that we're just missing them because we're not actually able to pick them up. So this is data from a set of experiments that we ran during our Hi-Fi Cas9 development to assess the off-target activity. So here we're testing a guide targeting AMX1 combined with a wild-type Cas9 or Hi-Fi Cas9 and delivered as a ribonucleoprotein. So beginning on the left-hand side here with the wild-type trial, so the orange bar on the is the percentage editing for the on-target assay. And this is designed to quantify the on-target edit. And here with our wild type, 98% of those reads map to the target. The pie chart, however, represents the total number of reads for all assays in the panel. And we can see that in the wild type trials, both trial A and trial B, 40% of all reads are 40% of all edits are close to off target. So when we look at our high fidelity Cas9, and we can see a different story, that it's a much cleaner system and almost 100% of all reads are going to the on-target assay. We've essentially all but eliminated off-target edits in this test. This is a similar experiment, but has been expanded to test more guides and slightly different conditions. So if we run here from the right-hand side of this chart, going down, we can see we have the androgen receptor, EMX1, HBB, and HPRT. And running along the bottom are some of the different conditions that we have. We have a wild-type Cas9 and is being expressed in a cell line. We have a high-fidelity Cas9 being expressed in a cell line. And then we have wild-type Cas9 delivered as a protein. and high fidelity Cas9 delivered as a protein. And we wanted to test all of these guides and assess their total editing depending on the method of Cas9 delivery. So if we start by looking at a wild type Cas9 expressing cell line for HPRT down in the very bottom left square, we can see that we have 72% on target editing for this particular one, and with around 28% off target editing. Simply by switching to an RNP, if you shift 2 boxes across to the wild type Cas9 ribonucleoprotein delivery, we can see that our off target editing has dropped to just 2%. And when we switch to an RMP for our high fidelity Cas9, we don't actually gain much. We gain a further 1%. But the change is specific to certain target sites. For example, if we look at EMX1 and we look again at the wild type Cas9 expressing cell line, you can see that 57% off target editing here as quantified by rhAmpSeq. And by switching to just a ribonucleoprotein and delivering the same protein, but not expressed in the cell line, we can make massive gains. But again, when we switch to a high fidelity Cas9, we are actually making significant gains this time. We've done this across many different guides, and we've used rhAmpSeq as our weapon of choice to analyze these samples. So that all the data I was showing in the previous slides was looking at the level of non-homologous end joining in CRISPR. And we also know that homology-directed repair is also occurring, and this happens. And so instead of joining the double-stranded breaks, a donor template is added, and this will have homology to either side of the cut site, and this can be incorporated during the repair process, and you can actually insert specific mutations or insertions on the donor template. And we can use rhAmpSeq also to assess the level of HDR. And that is exactly what we've done in this experiment. So we've looked at a few different guide sites here: AAVS1, MET, MYC, SAA1, Bruce 26, and TRAC. And we've done it in GERCAT and H3612J cell lines. We've delivered our donor as either an unmodified single-stranded oligo, an oligo which has phosphorothioate modified ends, and then we have our IDT modified donor. And you can see here highlighted in blue is the portion that we're trying to insert. And we can see that across all of the different guide sites and cell lines that it's consistently that the IDT alt-R modified donor template is getting the highest level of HDR. But again, it is variable by target site, whereas AAVS1 we're getting over 20% HDR, whereas in ROSA 26 that may be 6 or 7%. So it is variable and is dependent on the target site as well. And we have quantified the percentage homology directed repair using rhAmpSeq. So I just want to finish by summarizing some of the key features and benefits of rhAmpSeq. So we have two protocols, as I've mentioned. We have our regular and our high throughput protocol. The input range will depend on which protocol you choose, but generally from 10 to 100 nanogram. You can go lower, but this is the recommended input amount. At IDT, we've tested this on various sample types from tissue, FFPE, and cell-free DNA. Our insert size can be tuned from 50 to around 200 bases. If you want to get all of the information surrounding the site, you can sequence surrounding, or if you're just interested in a SNP, you can save on sequencing costs. We'll work with you to tune your insert size. Right now, we're topping out at 5,000 amplicons per panel. Our sample indexing capability is 96 UDIs or 9,000 combinatorial dual indexes. If you require more than 96 UDIs, we certainly have those barcodes and we can make them as custom oligos for you. These are the performance guarantees we're going to put on rhAmpSeq. And as I mentioned earlier, there are no stocked panels. Everything will be custom and all the design will be handled by IDT. So it's quite important that we put some performance guarantees on that, and these are some pretty good performance metrics that we will regularly meet. Right now, it's only compatible with Illumina platforms. We hope to support other instruments in the future, but right now it's just Illumina. And as I mentioned, we support our regular and high throughput library prep methods. If you're interested in learning more about rhAmpSeq, or you want any resources or any of the papers I've referred to, you can visit www.idtdna.com/rhAmpSeq. Thanks for tuning in to today's presentation.
Thank you, Simon, for that informative presentation. To the audience, if you have a question and have not done so already, please type it into the Q&A box. Okay, there are a few questions for you, Simon. Here's the first one. Let me push it to the slide area so that everybody could read that. Okay, the first one, can I use degenerate primers with rhAmpSeq?
Yeah, so we can make primers with degenerate bases in them, and there is a limit to how many runs of N bases we can have in there. But yeah, certainly we can use degenerate primers with rhAmpSeq. Okay, thank you, Simon. Just a level of degeneracy, but that's a really good question. And if you wanted to get a design, we'll get your details after this, and we'll contact you. Okay, sure. Here's another question. Let me push it out. Okay, are you able to design primers for amplicons or targets with not well characterized or established reference genomes? Sure. So we can. So we can design off the closest reference genome. It's always ideal to have a reference. If we don't, we can certainly design to just a FASTA file. It just limits our ability to perform all of our QC checks that we normally do. So if we don't have a complete reference to work off, we can certainly still do it, but just can't perform all of those QC checks that we normally follow. So yeah, another great question. Thank you there. Let's have another question coming up here. Right, what's the maximum number of targets can you do with rhAmpSeq? Okay, yeah, so as I mentioned, it's 5,000 targets at the minute. Okay. So if you needed more than that, you could do them in separate pools and mix them together for sequencing. But right now it's 5,000. But if anybody wants to do more than that, we're certainly willing to work with you. Mm-hmm. All right. There are 5,000 amplicons. Certainly a very mind-blowing number. Here's another question. Do you have more than 96 unique dual indexes? 96 is what's available on the website, but if you need more, we have access to a lot more indexes, and we can just give you the sequence of the oligo, and you can order it and put the extra index in there. So I think for 8 base pair indexes, we have up to 384, and if you wanted to do 10 base pair indexes, then we could go up to a couple of thousand. Okay, here's another question. How many reads do you need for each target? Okay, so that depends a little bit. So to accurately call genotypes, we recommend a minimum of 20 reads. That's in diploid species with heterozygosity, so i.e. 50% allele frequency. So for polyploid species or somatic mutations with lower allele frequencies, your minimum coverage will need to be higher. Okay. Here's another question. Why is there a 200 base pair limit on amplicon size? Can I target 500 base pair amplicons? With the design at the minute, we've decided to do shorter amplicons. We have to have a minimum distance in between amplicons as well, and the larger amplicons don't amplify as efficiency. So there's a couple of moving parts there. We certainly can do 500 base pair amplicons. We have worked with customers on larger designs. Again, it will depend how far apart the targets are as well, because we need at least 600 bases in between targets for this version of rhAmpSeq. But yeah, we try to keep it under 250. 500 base pair amplicons don't amplify as efficiently as well. But if that particular person wants to look at a design for larger amplicons, we will certainly work with them. Okay, thank you, Simon.
Here's another one. I think we're down to the last two questions that we have for today. Should all primer sequences be removed for sequencing data analysis?
Yes, so if you don't clip your primer sequences, it will mess with your alignment, and yeah, the reads won't align properly to your genome. So yes, definitely should be removed. There are a number of free tools to do that for you.
Mm-hmm. And finally, the last question for today. That's all the time we have for questions. Oh yes, this. Sorry if you've already mentioned this. I joined the webinar a couple of minutes late. Does the portion of primer to the 3' of the RNA base match the template?
Yeah, so it has to match the template because the RNase H2 enzyme is sensitive to mismatches around the RNA base. So therefore the portion after the RNA base also matches the template.
Right. Thank you. Thank you, Simon. That's all the time we have for questions. I want to thank all of you for attending today's presentation. I also would like to thank Simon for his informative presentation, as well as the question and answer session. This is one of a series of webinars we will be presenting on NGS, as well as other topics. We will email you about these future webinars as they are scheduled. Also, as a reminder, a recording of this webinar will be posted shortly on our website and at www.youtube.com/IDTDNABio. There you will find several other educational webinars on such topics such as next generation sequencing, genotyping, qPCR, and general molecular biology. Thank you, everyone. Thank you all for attending this webinar, and we wish you the best of success in your research.